my name is Strowig and I'm heading the Department of Microbial Immune Regulation at the Helmholtz Center for Infection Research in Braunschweig. But I'm also a professor for infection biology at the medical school in Hanover. Our research is about the human microbiome, so those microbial communities that colonize all surfaces of the human body. The most of these microorganisms are actually located in the gastrointestinal tract, where they are closely interacting with the mucosal immune system. So many associations have been described between changes in the composition of the microbiota and various diseases, but it's frequently not known whether these changes in the composition are actively or directly causing the disease or modulating the disease. So this is our daily business, to understand how those communities potentially contribute to the initiation of the diseases. Within the Excellence Cluster Resist, we are involved in two projects. One of them is working together with a rheumatologist, trying to understand whether the human gut microbiota drives and causes maybe arthritis. And this is a inflammatory disease of the joints with genetic causes, however also unknown environmental factors. Since many of the patients actually do not respond also to the initial treatment that the doctors prescribe them, we are also interested on whether, looking at the microbiota, we can predict whether the patients respond or do not respond to the medications to potentially improve the clinical practice. We are also involved in a second project where we, together with the pediatricians, are looking whether probiotics can be utilized to prevent enteric infections and sepsis in preterm children. So babies that are born too early and who are a particularly vulnerable group of patients. In the past, in our research, we had mainly focused on model systems in the laboratory, such as animals or cell culture systems. However, now within the Cluster of Excellence Resist, we are able to interact with the clinicians that are treating patients on a daily basis. And together with them, we are able to analyze samples from the patients in the context of the disease development, trying to improve how patients are treated on a daily basis. I'm most fascinated about how the interaction of these microbial communities happens with the host. So we have to imagine these are tiny organisms interacting with each other, with the immune system, and to really understand this interplay, we have to apply many different methods. We have to look at many different angles and have to apply concepts from ecology to microbiology to immunology. So it's a highly integrative work, and this is what really drives me on a daily basis.